Immunology, Chapter 17. Let us go. So here we will discuss general principle of transplantation, immunology, immune response to allograft, nature of allogens, the recognition of alloantigen by T cell, activation and effector function of allogens, T cells, activation of alloreactive B cells, and production and function of alloantibodies, innate immune response to allograft, pattern and mechanism of allographic rejection. I will discuss prevention and treatment of allographic rejection. We'll discuss about xenogenic transplantation, blood transfusion, and ABO, and also RH. This is rhesus group antigens. We'll discuss about hematopoietic stem cell transplantation, indication, method, and immune barriers. So it's like we are putting in practice the immunology again. So without further ado, let us go. Transplantation is widely used for replacing non-functioning organs and tissues with health organs or tissues. Transplantation is the process of taking cells, tissue, or organs called a graft from one individual and placing them into usually a different individual. The individual who provides the graft is called a donor, and an individual who receives the graft is called either the recipient or the host. If the graft is placed into its normal anatomic location, the procedure is called orthotopic transplantation. If the graft is placed in different site, the procedure is called heterotopic transplantation. Transfusion refers to the transfer of circulating blood cell or the plasma from one individual to another. Clinical transplantation to treat human disease has increased steadily during the past 60 years. And transplantation of hematopoietic stem cells, kidney, liver, and heart is now common practice in clinical medicine and transplantation of other organs such as lung and pancreas is becoming more frequent. We see in figure 17.1. Almost 40,000 kidney, heart, lungs, liver, pancreas, and the intestine transplant are currently performed in the United States each year. Transplantation of hands and faces are also now performed in the new medical centers, and transplantation of many other organ or cells, including tissue stem cells, is being attempted. After the technical challenge of surgical transplantation organ was overcome, it soon became clear that the immune response against grafted tissue is the major barrier to survival of transplanted tissue or organs. Conversely, controlling this immune response is the key to successful transplantation. This realization has led to the development of transplantation immunology as a discipline within the broader topic of immunology, and this is the theme for this chapter. General principle of transplantation immunology. Based on experimental studies and clinical observation, there are several principles that uniquely apply to immune response against transplants. Transplantations of cells or tissue from one individual to a genetically non-identical individual invariably lead to the rejection and of the transplants because of an adaptive immune response. This problem was first appreciated when an attempt to replace a damaged skin on burn patient with skin from the unrelated donor proved to be uniformly unsuccessful. Within 1 to 2 weeks, the transplanted skin would undergo necrosis and fall off. The failure of the graft led Peter Medawar and other investigators to study skin transplantation in the animal model. This experiment established that the failure of the skin graft was caused by inflammatory reaction, which was called a rejection. The knowledge that graft rejection is the result of an adaptive immune response came from experiment demonstrating that the process had characterized of memory and specificity and was mediated by lymphocyte. For instance, rejection occurred 10 to 14 times after the first transplant from a donor to a non-identical recipient called the first set rejection, and more rapidly after second transplant from the same donor to this recipient called second set rejection, implying that the recipient developed a memory for the graft tissue. Individuals who have rejected a graft from one donor shows accelerated rejection for another graft from the same donor but not from a different donor, demonstrating that the rejection process is immunologically specific. These experiment results were recapitulated in clinical transplantation, perhaps the most compelling evidence showing that allograft rejection is an adaptive immune response was finding that ability to rapidly reject a transplant with second set kinetics can be transferred with the lymphocyte from a transplant recipient's animal to a naive animal. So this is what have been happening. Number of solid organ transplant in the United States, as you can see. Kidney, it have been more relevant, followed by liver, followed by heart, followed by lungs, followed by pancreas, and followed by intestines more. So transplant immunologists have developed a special vocabulary to describe the kind of cell and tissue encountered in a transplant setting. A graft transplanted from one individual to the same individual is called the autologous graft. A graft transplanted between two genetically identical individuals is called the synergistic graft. A graft transplanted between two genetically different individuals of the same species is called the allogenic graft, allograft. A graft transplanted between individuals of different species called the xenogenetic graft or xenograft. The molecules that are recognized as foreign in allograft are called alloantigens, and those in xenograft are called the xenoantigens. The lymphocyte and the antibody that react with the alloantigens or xenoantigens are described as being alloreactive or xenoreactive, respectively. Most of this chapter focuses on allogenic transplantation because it's far more commonly practiced than xenogenetic transplantation, which is discussed briefly at the end of the chapter. We will consider both the basic immunology and some aspect of the clinical practice of the transplantation. We will conclude the chapter with the discussion of HS, hematopoietic stem cells transplantation, which raises a special issue not usually encountered with solid organ transplant. Immune response to allograft. Immune response that mediate allograft rejection are fundamentally similar to response that fight microbes. The main difference is nature of what is recognized as foreign. Adaptive immune response play the major roles in graft rejection, but innate response may contribute in some situation. In this section, we focus on adaptive immune response to alloantigens and mention the possible role of innate immunity later. Alloantigens list both cellular and humoral immune responses. The mechanism of allorecognitions are best understood by considering the graft antigen that stimulate allogenic response and the properties of the responding lymphocytes. The nature of alloantigens. Most of antigens that stimulate adaptive immune response against allograft are protein encoded by polymorphic genes that differ among individuals. These proteins are called histocompatibility molecule because they determine if the grafted tissue is compatible or incompatible with host immune system. The most important of these molecules are major histocompatibility complex proteins, already discussed in chapter 6. All of the animals of an inbred strain are genetically identical and they are homologous for all genes except genes on the sex chromosomes in males. In contrast, the inbred animals of different strains and individuals in an outbred species except identical twins differ in many of the genes they inherit. The basic role of transplantation immunology, which were first established from the experiment done with genetically defined mice, include the following. First, cell or organ transplanted between genetically identical individual, identical twins, or member of the same inbred strain of the animal are not rejected. Cell or organs transplanted between genetically non-identical people or member of the two different inbred strains of the species are almost always rejected. So these are just figures showing the first set and second set allogenic rejection that we said already that second set is mostly rapidly rejected. The offspring of the mating between two different inbred strains of animal will not reject graft from either parents. In other words, an A plus times B first filial generation animal will not reject graft from an A and or B strain animal. This rule is violated by hematopoietic stem cell transplantation when a natural killer cell is in A times B first filial generation recipient to reject the HSCs from either parent or as well as we will discuss later in this chapter. A graft derived from offspring of mating between two different inbred strains of animal will be rejected by either parents. In other words, graft from an A times B first filial animals will be rejected by either A or B strain animal. Such results indicate that the molecule in the graft that are responsible for eliciting rejection must be polymorphic and their expression is concomitant. Concomitants. Polymorphic refer to the property that these graft antigens differ among the individual of the species other than identical twins or between different inbred strains of animals. Codominance expression means that every individual inherits a gene encoding this molecule from both parents, and both parents' alleles are expressed. Therefore, A and B animal express both A and B alleles. These see both A and B tissue as self and are tolerant to both A and B proteins. By contrast, the inbred A and B animal expresses only that allele and are not tolerant to protein they do not express and see A and B tissue as partly foreign. Thus, A and B animal does not reject either A or B strain graft, whereas both A and B strain recipients reject A and B falciparum graft. So you will see donor strain B, donor strain A, donor strain B, donor strain B, donor strain A and B. So donor strain A, graft recipient strain A, no rejection. Synergetic graft, no rejection. Donor, this is strain B, is taken the tissue, recipient is recipient strain A, then this is recipient full allogenic graft is rejected. Donor strain B, then the recipient is AB, no rejection. Then donor strain AB, then the recipient is A, tissue is rejected because there are some kind of new thing. So the genetic of graft rejection in the illustration, the two different mouse color represent inbred strain with different set of genes referred to here as A and B, encoding molecular importance of graft rejection called the major histocompatibility complex molecule. Inherited major histocompatibility complex allele from both parents are codominantly expressed in the skins of an A times B offspring, and therefore these mice are represented by both color. Synergic graft are not rejected. Allograftic are always rejected. B. Graft from an A or B parent will not be rejected by an A-B phosphorylase offspring, but graft from the first filial will be rejected by either parent. These phenomena are due to the fact that MHC genes products are responsible for graft rejection, Grafts are rejected only if they express an MHC types represented by green or orange that is not expressed by the recipient mouse. The genetics of graft rejections provide one of the earliest lines of evidence that adaptive immune system distinguish itself from foreign antigens. All individuals are normally tolerant to self-antigens and usually not react against these, but they do react against foreign antigens. This is now known to be fundamental property of normal immune system, thereby foreign most often refer to microbial agents. In the context of organ transplantation, foreign usually means product of polymorphic MHC genes. The molecule responsible for strong and rapid rejection reactions are MHC molecules that bind and present peptide to T cells. MHC molecules described in chapter 6 were named before their physiological function was understood. George Snell and colleague produced pair of congenic strain of inbred mice that were bred to genetically identical to each other except for gene needed for graft rejection. They used these mice to identify the polymorphic genes which were called MSC genes that encode the molecular targets of allographic rejection. The transplant of the most tissue between any pair of unrelated individuals will be rejected because genes encoding MSC molecule are so polymorphic that two unrelated individuals are extremely unlikely to inherit the same alleles. The role of MSC molecules as antigen that cause graft rejection is consequence of the nature of T cells antigen recognition, as we'll discuss later. Recall that human MSC molecules are called human leukocyte antigens, and in the context of human transplantation, the terms MHC and MHL and HLA are used interchangeably. In any transplant between a genetically non-identical donor and a recipient, there will be a polymorphic antigen other than MHC molecule against which the recipients may mount an immune response. This antigen typically induce weak or slower, more gradual rejection reaction that do MHC molecule and therefore that they will, let me repeat, sorry. These antigen typically induce weak and slow, more gradual rejection reactions that do MHC molecules and therefore called minor histocompatibility antigen. The relevance of the minor histocompatibility antigens in clinical solid organ transplantation is uncertain, mainly because there have been a little success in identifying the relevant antigen. In mice, the male HY antigen appear to be a target of the immune recognition of female recipients of graft from male donor. Although in humans there is a slightly higher risk of rejection of heart transplant from male donor to female recipients compared to with gender matched transplant, given the scarcity of the donor heart, gender matching is not practical. Minor histocompatibility antigen play a more significant role in stimulating graft versus host response after HS, HSC transplantation, discussed later. But the nature of the relevant antigen in that setting is also not defined. So just these ones are the diagram. Direct allogenic recognition, allogenic MHC, alloreactive CD4, T cell cytokines, T cell recognized donor MC bond peptides, then the graft rejection happen. Indirect allogenic recipients happen, as is told it is slow. So the slow and the rapid they can happen. So the direct and indirect allogenic recognition, this is the figure 17.4. Direct allogenic recognition occur when alloreactive T cell bind directly to intact allogenic image histocompatibility complex MHC molecule with bond peptide on or grafted donor dendritic cells or other antigen presenting cells within Lymph nodes. Recipient CD4 or CD8 T cells can directly recognize donor class 2 or class 1 MHC molecule respectively and will differentiate into helper T cells and or cytotoxic T lymphocyte. The cytotoxic T lymphocyte will directly recognize the same donor MHC peptide complex displayed on graft tissue cells and kill these cells. B. Indirect allogenic recognition occurs when allogenic MHC molecules from graft cells are taken up and processed by recipient antigen-presenting cells and peptide fragments of allogenic MHC molecule containing polymorphic amino acid residue are bound and presented by recipients self-MHC molecules. Donor MHC specific helper T cells that, that are generated in this way can help B cells to produce donor MHC antibodies that can damage graft cells. The helper T cells also can be activated in the graft by recipient's macrophage presenting the same donor MHC-derived peptide leading to inflammatory damage of the graft. So recognition of alloantigen by T cells. Allogenic MHC molecules of a graft can be presented by recognition by recipient T cells in two different ways, called direct and indirect. Initial studies show that T cells of graft recipients recognize intact and processed MHC molecule in graft, and this is called direct presentation or direct recognition of alloantigens. Subsequently, studies show that sometimes the recipients of T cells recognize graft donor MHC molecule only in the context of recipient's MHC molecule, implying that the recipient's MHC molecule must be presented peptide derived from allogenic donor MHC protein to recipient T cell. This process is called the indirect presentation or indirect recognition, and it is essentially the same as the recognition of any foreign microbial proteins antigen. The initial T cells response to MHC antigens, whether it results from direct or indirect recognition, most likely occur in lymph node draining the graft, as we'll discuss later. Direct recognition of major histocompatibility complex alloantigens on donor cells. In the case of the direct recognition, intact MHC molecule displayed by cells in the graft are recognized by recipient T cell without a need for processing by host antigen presenting cell. It may seem puzzling that T cells that are normally selected during their maturation to be self-MHC restricted are capable of recognizing foreign, allogenic or xenogenic MHC molecule. A likely explanation is that T cells receptor have some intrinsic affinity for MHC molecule, regardless of whether they are self or foreign. Furthermore, during T cell development in thymus, positive selection promotes survival of T cells with weak self-MHC reactivity, and among these T cells, there are may be many with strong reactivity to allogenic MHC molecules. Negative selection in the thymus efficiently eliminates T cells with high affinity to self-MHC, already discussed in chapter 8 and chapter 15. But it will not eliminate T cells that bind strongly to allogenic MHC molecules simply because allogenic MHC molecules are not present in thymus. The result is that the mature repertoire includes many T cells that bind allogenic MHC molecule with high affinity. One can think of different allorecognition as an example of immunologic cross-reaction in which a T cell that was selected to be self-MHC restricted is able to bind structural similar allogenic molecule with sufficiently high affinity to permit activation of T cells. MHC molecules that are expressed on the cell surface normally contain bound peptide, and in some cases the peptide contribute to the structural recognition of alloreactive T cells. exactly like the roles of peptide in the normal recognition of foreign antigen by self-MHC-restricted T cells. Even though these peptides may be derived from proteins that are present in both donor and recipients, on the graft cells they are displayed by allogenic MHC molecules. Therefore, the complexes of peptide self or foreign with allogenic MHC molecule will appear different from self MHC complexes. In other cases, direct recognition of and active direct recognition and activation of an alloreactive T cells may occur regardless of which peptide is carried by allogenic MHC molecule, because the polymorphic amino acid residue of the allogenic MHC molecule alone form a structure that can resemble self-MHC plus peptide. T cell response to directly presented allogenic MHC molecule are very strong because there is a high frequency of T cell that can directly recognize any single allogenic MHC protein. It is estimated that as many as 1% to 10% of all T cells in an individual will directly recognize and react against allogenic MHC molecules on a donor cells. In striking contrast, in an infection, the frequency of naive T cells that react against any microbial peptide displayed by a self-MHC molecule is approximately 1 in 10 power 5 to or 10 power 6 T cells. There are several explanations for high-frequency alloreactive T cells. Many different peptides derived from donor cellular proteins may combine with a single allogenic MHC molecule, and each of these peptide MHC combination can theoretically activate a different clone of recipient T cells. In contrast, most microbes or protein antigen contain relatively few immunodominant peptides that can be displayed by self-MHC molecules of an individual at any time, so few T cells clones are activated. It is estimated that each antigen presenting cell expresses thousands of MHC molecules. On an allogenic antigen presenting cell, most of these can be recognized by reactive T cells at one time. However, in case of infection, less than 1%, and perhaps as few as 0.1% of self-MHC molecules on antigen-presenting cells normally present peptide from that microbes at one time, and only these can be recognized by T cells specific for microbial antigen. Allogenic MHC molecule can display not only foreign peptides from donor cells but also self-peptide, and this self-peptide foreign MHC complex can activate T cells. Because these complexes are not normally expressed in thymus or peripheral tissue, they have not participated in negative selection of T cell that could recognize allogenic graft cells and potential cause of graft damage. In contrast, T cells specific for self-peptide displayed by self-MHC molecules are eliminated by negative selection in thymus and by peripheral tolerance mechanisms. Therefore, the range of peptide MHC complexes that can activate T cells is much greater if the MHC is allogenic. Many of the T cells that respond to an allogenic MHC molecule, even first exposure, are memory T cells. It is likely that these memory cells are generated during previous exposure of other foreigns, e.g. microbial antigen, and cross-reactive with allogenic MHC molecule. These memory cells not only expanded population of antigen-specific cell, but also more rapid and powerful responder than a naive lymphocyte. Thus, these alloreactive memory T cells contribute to the greatest strength of initial T cell response to new allograft compared to the response to the first encounter with microbial antigen. Direct recognition can generate both CD4 and CD8 T cells that recognize graft antigen and contribute to rejection. The role of alloreactive T cells response in rejection is described later. Indirect recognition of alloantigens. In the indirect pathway, donor allogenic MHC molecules are captured and processed by recipient antigen-presenting cells and peptide derived from the allogenic MHC molecules are presented in association with self-MHC molecules. Thus, the peptide from allogenic MHC molecule are displayed by host antigen-presenting cells and recognized by T cells like conventional microbial protein antigen. Because allogenic MHC molecules have amino acid sequence different from those of the host, they themselves can serve as foreign antigen and generate foreign peptide associated with self-MHC molecules on the surface of host antigen presenting cell. Each allogenic MHC molecule may give rise to the multiple peptides that are foreign for the host, each recognized by a different clone of T cell. Indirect presentation may result in recognition by CD4 T cells because alloantigen are acquired by host antigen presenting cells primarily through endosomal vesicular pathway. This is a condenser of the phagocytosis and therefore presented by a class 2 MHC molecule. Some antigen of the phagocytosed graft cells do enter the class 1 MHC pathway of antigen presentation and are indirectly recognized by CD8 T cells. This phenomenon is an example of cross-presentation or cross-priming, in which dendritic cells ingest a protein of another cells, such as from the graft. The proteins are derived to cytosols where they are processed into the peptide by proteasomes, and the peptides are presented on class 1 MHC molecule to activate prime CD8 T cells. Evidence that indirect recognition of allogenic MHC molecule play a significant role in graft rejection come from the studies with knockout mice lacking the class 2 MHC expression. For example, skin grafted from donor mice lacking class 2 MHC are able to induce recipient CD4, this is class 2 MHC restricted T cell response to peptide derived from donor class 1 MHC molecule. In those experiments, the donor class 1 MHC molecules are processed and presented by self class 2 molecule on recipient of antigen presenting cells and stimulate the recipient's helper T cells. Evidence also has been obtained that indirect antigen presentation may contribute to the chronic rejection of human allograft discussed later. The CD4 T cells from heart and liver allograft recipients recognize and are activated by peptide derived from donor MSC when presented by the patient's owner antigen presenting cell. The indirect pathway may become more important over time after transplantation as donor dendritic cells in the graft are replaced by host donor dendritic cell. Activation and effector function of alloreactive T lymphocyte. When lymphocytes recognize alloantigen, they become activated and proliferate, differentiate, and perform effector function that can damage graft. The activation steps are similar to those we have described in the lymphocyte reaction in microbial antigen, the activation of alloreactive T lymphocyte. The T cells responsible to an organ graft may be initiated in the lymph nodes that drain the graft. Most organs contain resident antigen presenting cells such as dendritic cell, and therefore transplanted organs carry with them antigen presenting cells that express donor MHC molecule. These donor antigen presenting cells can migrate to regional lymph nodes and present on the surface and process allogenic class 1 or class 2 molecules to recipient CD8, CD4 T cells, respectively. Direct MC allo recognition. Host dendritic cells from the recipients may also migrate to the graft, pick up a graft allogenic alloantigens and transport these back to the draining lymph nodes, there where they are displaced the indirect pathway. The connection between lymphatic vessel in the allograft and recipient lymph nodes is surgically disrupted during the process of transplantation, and it's likely reestablished by growth of the new lymphatic channel in the response to inflammatory stimulus produced during grafting. Naive CD4 and CD8 lymphocytes that are normally traffic through the lymph nodes encounter these alloantigen and are induced to proliferate and differentiate into effector helper T cells and cytotoxic T lymphocytes, CTLS. This process is sometimes called sensitization to alloantigens. The effector cell migrate back into the graft and immediate rejection by mechanisms that are discussed later. As mentioned earlier, many of the T cells that respond to alloantigenic MHC antigen is in a new graft are cross-reactive memory T cells previously generated against the environmental antigen before transplantation. Unlike naive T cells, memory T cells may not need to see antigen presented by dendritic cells in lymph node to be activated. They may migrate directly into graft where they can be activated by antigen presenting cell or tissue cell displaying alloantigens. So this is a figure 17.6, activation of alloreactive T cells. A, they say this is kidney, allograft kidney. A is in case of the direct allorecognition, donor dendritic cells in the allograft migrate to the secondary lymphoid tissue where they directly present allogenic major histocompatibility complex molecules to host T cells. Only CD8 T cells recognizing donor class 1 MHC is shown, but CD4 T cells can also directly recognize donor class 2 MHC. In the case of indirect allorecognition, recipients' dendritic cells that have entered the allograft transplant donor MHC protein to secondary lymphoid tissue and present peptide derived from these MHC protein to alloreactive host T cells. This is shown as CD4 T cells and indirect recognition of allogenic MHC by CD8 T cells likely less important. After both indirect and direct allorecognitions, the T cells become activated and differentiate into effector CD4 and helper T cells and the CD8 cytotoxic T lymphocytes. In B, the alloreactive effector T cells migrate into the allograft, become reactivated by alloantigen and mediate damage. In the graft, direct recognition of allogenic class 1 by CD8 CTLS is required for killing of graft parenchyma cells because these cells express only allogenic MHC. In contrast, CD4 helper T cells that can directly or indirectly recognize allogenic class 2 MHC can be activated by donor or host antigen presenting cells, respectively, and both can promote inflammation that damage the graft. So this is the diagram. If you follow it, this allographic kidney, inside there is donor dendritic cells, the recipient dendritic cells, they have come in. These are called donor alloantigens. So transport of alloantigens to lymph nodes, then afferent lymph nodes, you see here after the transfer here, activation of T cell, generation of effector T cells by direct and indirect antigen presentation will take place. Then from here, recipient CD8 effector cells and also other cell effector lymph nodes, it will transport back recipient effector T cell migrations. They will migrate back. When they come back here, then recipient's antigen presenting cell activation of effector T cells, biologenic graft rejection, then cytokines killing of graft tissue will take place. So the response of alloreactive T cell to foreign MHC molecule can be analyzed in vitro by mixed lymphocyte reactions, in which lymphocytes from two genetically distinct individuals are mixed together in cell culture. The T cells from one individual become activated by recognition of allogenic MHC molecules on the cell of others. The mixed lymphocyte reactions was used clinically in the past as a predictive test of T cell mediated graft rejection and as an in vitro model to study the mechanism of alloreactives, but now it's mainly a historical significance. Role of co-stimulation in T cell response to alloantigens. In addition to recognition of alloantigens, co-stimulation of T cell primarily by B7 molecules on antigen-presenting cell is important for activating alloreactive T cells. Co-stimulation is likely most important to activate naive alloreactive T cell, but even alloreactive memory T cells response can be enhanced by co-stimulation. Rejection of allograft and the stimulation of alloreactive T cells in MLR can be inhibited by agents that block B7 molecules. Allograft survive for longer period when they are transplanted into knockout mice lacking B7A, CD8, and the B72, CD86 compared with the transplant in normal recipient. As we will discuss later, blocking B7 co-stimulator is a therapeutic strategy to inhibit graft rejection in animal, in mammal, in human as well. The requirements for co-stimulation lead to the interesting questions of why these co-stimulators are expressed by graft antigen presenting cells in absence of infection, which we have previously discussed in the physiological stimulus of expression of co-stimulators. One possibility is that the innate immune response to ischemic damage of some cells in graft, discussed later, result in increased expression of co-stimulator on antigen-presenting cells. Effector function of alloreactive T cells. Alloreactive T cells, alloreactive CD4 and CD8 T cells that are activated by graft alloantigens cause rejection by distinct mechanism. We'll see them in figures 7-6 already have discussed. The CD4 helper T cells differentiate into cytokine-producing effector cells that damage graft by cytokine-mediated inflammation, are similar to a delayed type hypersensitivity reactions. See in chapter 10 and chapter 19. CD8 T cells are differentiated into cytotoxic T cell, which kills graft cells. Only cytotoxic T lymphocytes that are generated by direct allorecognition can kill graft cells, whereas both CTL and helper T cells generated by either direct or indirect alloantigen recognition can cause cytokine-mediated damage to graft. CD8 cytotoxic T cells that are generated by direct allorecognition of donor MHC molecules, donor antigen present can recognize the same MHC molecules on parenchyma cells in the graft and kill those cells. These T cells that can also secrete cytokine that can cause damage inflammations. In contrast, NSCD8 T cells, cytotoxic T cells that are generated in response to indirect recognition of allogenic MHC are restricted to recognition of peptide from these allogenic MHC molecule bound to recipient self MHC molecule, and therefore the T cells will not be able to kill the foreign graft cells because the graft cell do not express recipient MHC molecules. When CD4 effector T cells are generated by direct or indirect recognition of allogenic MHC, the principal mechanism of rejection is inflammation caused by cytokine produced by effector T cells. CD8 T cells that may be activated by the indirect pathway may also contribute to the rejection by producing inflammatory cytokines. Presumably, effector cells activated by indirect pathway infiltrate the graft and recognize peptide from graft MHC molecule being displayed by host antigen presenting cells that have also entered the graft. Activation of alloreactive B cells and production and function of alloantibodies. Antibodies against graft antigens, called donor-specific antibodies, also contribute to the rejection. Highly affinity alloantibodies are mostly produced by helper T cell dependent activation of alloreactive B cells, much like antibody against other protein antigens. The antigen most frequently recognized by alloantibodies are donor MHC molecules, including both class 1 and class 2 MHC protein. The likely sequence of events leading to the generation of these alloantibodies producing cell is that naive B cells lymphocyte recognize the allogenic MHC molecule, internalize and process these proteins, and present peptide derived from them to help a T cell that are previously activated by the same peptide presented by dendritic cells. Thus, activation of alloreactive B cells is an example of indirect presentation of alloantigens. In addition, donor-specific antibody against non-HLA alloantigens also contribute to rejection. The alloreactive antibody produced in the graft recipients engage the same effector mechanism that antibodies use to combat infection, including complement activations and AFC receptor-mediated binding and activation of neutrophil, macrophage, and natural killer cells. Because MHC activation antigens are expressed on endothelial cells, much of the alloantibody-mediated damage is targeted at graft vasculature, as discussed later. Innate immune response to allograft. In addition to the adaptive immune response specific for alloantigen, difference between donor and host innate immune play a role in the outcome of transplantation. The interruption of the blood supply to the tissue and organ during time between removal from the donor and placement in the host usually cause some ischemic damage. This can result in expression of damage-associated molecule pattern dumps in graft, which stimulate the innate response mediated by both innate cells within the graft and the recipient innate immune system. This innate response can directly cause graft injuries, but they are also thought to enhance adaptive response by increasing migration of alloreactive circulating memory T cells into the graft and by activating antigen-presenting cell, as in the case of immune response to microbes. Antigen-presenting activation increases the expression of costimulators and production of cytokines, and thus priming of naive alloreactive T lymphocyte. In addition, host natural killer cell can respond to the absence of self-MHC molecule on donor graft cells, therefore contribute to graft rejection. Pattern and the mechanism of allograft rejection. Thus far, we have described the molecular basis of alloantigens recognition and the cell involved in recognition and response to allograft. We now turn to the consideration of the effector mechanisms responsible for immunological rejection and allograft in different experimental model and in clinical transplantations. Alloreactive CD4 and CD8 T cells and alloantibodies all have been shown to be capable of mediating allograft rejection. These different immune effectors cause graft rejection by different mechanisms, and all three effectors may contribute to rejection concurrently. For historical reasons, graft rejection is classified on the basis of the time course of rejection after transplantation and histopathological features rather than immune effector mechanism. Based on the experience of renal transplantation, the histopathological pattern are called the hyperacute, acute, and chronic. It is helpful to consider rejection in context of this pattern because the immune mechanism of rejection correlates well with the pattern. Our discussion of these types of rejection will emphasize the underlying mechanism rather than the pathological or clinical features. Hyperacute rejection. Hyperacute rejection is characterized by thrombotic occlusion of the graft vasculature that begin within a minute to hour after host blood vessels are anastomized to graft vessels and mediated by pre-existing antibody in the host circulating that bind to donor and endothelial antigens. Binding of antibody to endothelium activates the complement, and antibody and complement product together induce number of changes in graft endothelium that promote intravascular thrombosis. Complement activation lead to the endothelial cells injuries and exposure of subendothelial basement membrane protein that activate platelets. The endothelial cells are stimulated to secrete high molecular weight forms of von Willebrand factor, which cause platelet adhesions and aggregations. Both endothelial cells and platelet undergo membrane vasculations, leading to shedding of lipid particles that promote coagulation. Endothelial cells lose the cell surface heparins, surface proteoglycan that normally interact with the antithrombin 3 to inhibit coagulation. These processes contribute to the thrombosis and vesicular occlusion, and the grafted organs suffer irreversible ischemic necrosis. So we see the structure. This can be hyperacute rejection, and in A is hyperacute rejection. Preformed antibody react with the vesicular endothelials. You see the antibodies activate complement and trigger rapid intravascular thrombosis and necrosis of vessel. In B, the hyperacute rejection involving glomerulus in the kidney allograft. Typical feature include endothelial damage and thrombi and leukocytic infiltration. So here we have complement activation, endothelial damage, inflammation, and thrombosis, cyclotin allogenic specific antibodies. In the early days of transplantation, hyperacute rejection was often mediated by pre-existing IgMs and alloantibodies, specifically for carbohydrate ABO blood group antigen that are expressed on red blood cells and endothelial cell. These natural antibodies are present in most individuals, discussed later. Hyperacute rejection by anti-ABO antibody is extremely rare now because all donors and recipients pair are selected so that they have compatible ABO types. Hyperacute rejection caused by natural antibodies specific for variety of antigen that differ among species is a major barrier to xenotransplantation and limit the use of animal organ for human transplantation. Currently, the rare instances of hyperacute rejection of allograft that do occur are mediated by IgG antibodies directed against the protein alloantigens such as donor MHC molecules or against less defined alloantigen expressed on vesicular endothelial cells. Such antibodies genetically arise as a result of previously exposure to alloantigens through blood transfusion, previous transplantation, or multiple pregnancies. In this level of these alloreactive antibodies is if the level of these alloreactive antibodies is low, hyperacute rejection may develop slowly over several days, but the onset is still earlier than typical for acute rejection. As we will discuss later, patients in need of allograft are routinely screened before grafting for presence of antibodies that bind to blood cells of a potential organ donor to avoid the use of organ that will likely suffer the acute rejection. In an unusual case in which graft have to be done between ABO incompatible donor and recipient, graft survival may be improved by rigorous depletion of antibody and the B cells. Sometimes, if the graft is not rapidly ejected, it survives even in the presence of antibody anti-graft antibodies. One possible mechanism for this resistance of hyperacute rejection is increased expression of complement regulatory proteins on graft endothelial cells, a beneficial adaptation of tissue called accommodations. Acute rejection. Acute rejection is the process of injury to the graft parenchyma and blood vessel mediated by alloreactive T cells and antibody. Before modern immunosuppression, acute rejection would often begin several days to few weeks after transplantation. The time of onset of acute rejection reflects time needed to generate alloreactive effector T cells and antibodies in response to graft. In current clinical practice, episodes of acute rejection may occur at much later times, even years after transplantation. If immunosuppression is reduced for any number of reasons, although the pattern of acute rejection are divided into acute T-mediated cellular rejection and acute antibody-mediated humoral rejection, both often coexist in organ undergoing acute rejection. Acute cellular rejection. The principal mechanism of acute cellular rejection are CTLA, cytotoxic T lymphocyte mediated killing of grafty parenchyma cells and endothelial cells, and inflammation caused by cytokine produced by helper T cells. On histological examination of kidney allograft, where these types of rejection is best characterized, there are an infiltrates of lymphocytes and macrophage. In kidney allograft, the infiltrate may involve tubuli called tubulitis, with associated tubular necrosis and blood vessel called endotheliolitis, with necrosis of the wall of capillaries and small arteries. The cellular infiltrate present in the graft undergoes acute cellular rejection includes both CD4 plus helper T cells and CD8 cytotoxic specific for grafty alloantigens, and both types of T cells may cause parenchyma cells and endothelial injuries. The helper T cells are mainly T helper 1 cells that produce interferons gamma and tumor necrosis factor. They cause macrophage and endothelial activation and inflammatory damage to the organs. Experimentally, adaptive transfer of alloreactive CD4 helper T cells and CD8 cytotoxic T cell can cause acute cellular graft rejection in recipients mice. So we have the diagram here. Acute figure 17.8. Acute cellular rejection. A is an acute cellular rejection. CD8, CD4, and CD8 T lymphocytes reactive with alloantigens on endothelial cells in blood vessel and parenchyma cells mediate a damage to those cell types. B. Acute cellular rejection of kidney with inflammatory cells in the connective tissue around the tubule and between epithelial cells of the tubules. C is inflammation of endothelial layer of blood vessel endotheliitis in acute cellular rejection with inflammatory cell damaging the endothelials. So if we look at here, you have a lot of macrophage. So A is you see there is endothelial cells, but they are antigen presenting cells here, and this one they cause being activated by these T cells because these are cells that are responsible. The macrophagy, the endothelial cells, you see they have activated these neutrophils. Think neutrophils. So parenchyma cell damage, interstitial inflammation, neutrophils. These are macrophage. Yeah. So the inflammation, as you see here. Acute antibody-mediated humoral rejections. Alloantibody cause acute rejection by binding to alloantigens, mainly HLA molecules, on vesicular endothelial cell, leading to endothelial injury and intravascular thrombosis that result in graft destruction. The binding of alloantibodies to endothelial cell surface trigger local complement activation, which causes lysis of the cells, recruitment and activation of neutrophils, and thrombus formation. Alloantibody may also engage Fc receptor on neutrophil and natural killer cells, which then kill the endothelial cells. In addition, alloantibody binding to endothelial surface may directly alter endothelial function by inducing intracellular signal that enhance surface expression of pro-inflammatory and pro-coagulants molecules. The histologic hallmark of acute antibody-mediated rejection of renal allograft are acute inflammation of glomerular and peritubular capillaries with focal capillary thrombosis. Immunohistochemical identification of C4D complement fragment in capillaries of renal allograft is used clinically as an indicator of activation of classical complement pathway and humoral rejection. Chronic rejection. As therapy for acute rejection has improved, The major cause of failure of vascularized organs allograft has become chronic rejection. Since 1990s, 1 year survival of kidney allograft has been better than 90%, but the 10-year survival has remained approximately 60% despite advance in immunosuppressive therapy. Chronic rejection develops insidiously during months or years and may or may not be preceded by clinically recognized episode of acute rejection. Chronic rejection of different transplanted organ is associated with distinct pathological changes. In the kidney and the heart, chronic rejection results in vesicular occlusion and interstitial fibrosis. Lungs transplants undergo chronic rejection shows thickened small airway called bronchiolitis obliterans, and liver transplants show fibrotic and non-functional bile ducts. So here is acute antibody rejection. So it's like continuation of what we saw last time is here, and it's continuation, I think. These are complement antibodies they will activate. So you can read for your own time. A dominant lesions of chronic rejection is in vascularized graft is arterial occlusion as a result of the proliferation of intimal smooth muscle cells, and graft eventually fail mainly because of resulting ischemic damage. The arterial changes are called graft vasculopathy or accelerated graft arteriosclerosis. Graft vasculopathy is frequently seen in failed cardiac and renal allograft and can develop in vascularized organ transplant within 6 months to a year after transplantation. The likely mechanism underlying the occlusive vesicular lesions of chronic rejection are activation of alloreactive T cells and secretion of uh, um, interferon gamma and other cytokines that stimulate proliferation of vesicular smooth muscle cells. As arterial lesions of graft arteriosclerosis progress, blood flow of the graft parenchyma is compromised, and parenchyma is slowly replaced by non-functional fibrous tissue. The interstitial fibrosis seen in the chronic rejection also may be a repair response to parenchyma cell damage caused by repeated bouts of acute antibody-mediated and cellular rejection. Perioperative ischemia, toxic effects of immunosuppressive drugs, and even chronic viral infection. Chronic rejection lead to the congestive heart failure or arrhythmias in cardiac transplant patients, or loss of glomerulus and tubular filtration and renal failure in kidney transplant patients. Prevention and treatment of allograft rejection. If the recipient of allograft has full functional immune system, transplantation almost invariably results in some form of rejection. The strategy used in clinical practice and in experimental model to avoid or delay rejection are general immunosuppressions and minimizing the strength of the specific allogenic reaction. An important goal for transplantation research is to find ways of inducing donor-specific tolerance, which would allow graft to survive without non-specific immunosuppressions. Method to reduce the immunogenicity of allograft. Solid organs used in transplantation come from both living and deceased donor, and graft survival after transplantation varies depending on the source. The greatest barrier to transplantation as therapeutic option for organ failure is the availability of organ. Currently in the United States, there are approximately 110,000 people people in need of life-saving organ transplant, but there have been only approximately 20,000 donors per year. Living donor can donate one kidney, a lobe of lungs, and part of the liver, pancreas, and intestine because they can remain healthy after these types of donation. Living donor may be genetically related to the recipients, including sibling, parent, children over 18 years of age, aunts, uncle, cousin, nieces, and nephews. Other living donor may be unrelated, as we have discussed. Immunological graft rejection is targeted at the allogenic proteins encoded by polymorphic allele in the recipient, not shared by donor. Related donor will share more allele of polymorphic genes, including MHC genes, than unrelated donor, and this will reduce the incidence and severity of rejection episode as discussed later. For example, because MHC genes are inherited as linked haplotypes, there is a 25 chance that two siblings will have identical MHC genes, whereas the chance of unrelated donor and recipient having identical MHC genes is extremely low. So this is chronic rejection. They are continuing with the diagram. Please get time ready for the diagram. Deceased donor called cadaveric donor are the source of transplantable organs and the only source of organ that could not be removed from living donor, such as heart. Most deceased donor are brain dead with complete and irreversible loss of higher brain function, but whose other organs can be kept alive in body by cardiorespiratory life support. Until just before organ harvest, less frequently organs are retrieved from people after very recent but irreversible cessation of circulation and respiration, such as after trauma. The survival of graft from deceased donor is on average lower than from either related or unrelated donor because there is more ischemic damage to organ removed after death of the donor. Furthermore, most deceased donors are unrelated to recipients, and graft from unrelated donor usually expresses more antigen that differ from those of the recipient and can stimulate stronger rejection response than those from living donor. In human transplantation, the major strategy to reduce graft immunogenicity has been to minimize alloantigenic difference between the donor and recipients. Several clinical laboratory tests are routinely performed to reduce the risk of immunological rejection of allograft. This includes ABO blood typing, determination of HLA allele expressed on the donor and recipient cells, called tissue typing, the detection of pre-performed antibodies in recipients that recognize HLA and other antigen representative of the donor population, and the detection of preformed antibody in recipients that bind to the antigen of an identical donor cells, called the cross-matching. Not all of these tests are done in all types of transplantation. We will next summarize each of these tests and discuss their significance. To avoid hyperacute rejection, the ABO blood grouping antigen of graft donor are selected to be compatible with the recipients. This test is uniformly used in organ transplantation because graft will typically not survive if there are ABO incompatibility between the donor and recipients. Natural IgM antibodies specific for allogenic ABO blood group antigen will cause hyperacute rejection. Blood typing is performed by mixing a patient's red blood cells with a standardized serum containing anti-A or anti-B antibodies. If the patients express either blood group antigen, the serum specific for that antigen will be agglutinated in red blood cells. The biology of ABO blood grouping system is discussed later in chapter in the context of blood transfusion. In kidney transplantation, the more MHC allele that are matched between the donor and recipient, the better the graft survival. HLA matching had more profound influence on graft survival before modern immunosuppressive drugs were routinely used, but current data still show significantly greater survival of graft when a donor and recipients have fewer HLA allele mismatch. Past clinical experience with older typing methods show that all class 1 and class 2 MHC logs matching at HLA-A, HLA-B, HLA-DR is the most important for predicting survival of kidney allograft. HLA-C is not as polymorphic as HLA-A, HLA-B, HLA-DR, and HLA-DQ are linked in this equilibrium, so matching at DR locus often also matches to DQ locus. Although current typing protocol in many centers include HLA-C, HLA-DQ, and HLA-DP lock, most of available data in the predicting graft outcome refer only to HLA, HLA-B, HLDR mismatch. because the two codominant expressed allele are inherited for each of these HLA genes. It is possible to have 0 to 6 HLA mismatch of these three lock between the donor and the recipients. Zero antigens mismatch predict the best survival of the living related donor graft, and graft with the one antigen mismatch are similar. The survival of graft with 2 to 6 HLA mismatch is significantly worse than that of the graft with 0 and 1 antigen mismatch. Mismatching of 2 or more HLA genes has an even greater impact on non-living unrelated donor non-renal allograft. Therefore, attempts are made to reduce number of difference in HLA allele expressed on the donor and recipient cells, which will have a modest effect in reducing the chance of rejection. So this is number of mismatch. If you have influence of major histocompatibility complex matching graft survival, matching the major histocompatibility complex alleles between the donor and the recipient significantly improve renal allograft survival. The data shown this for deceased donor cadaver graft. Human leukocyte antigen MHD has less impact on survival gene graft from live donor, and some MHC molecules are more important than others in determining outcome. So as you see here, 0. If there is 0 mismatch, the survival is high, almost 70 something, more than 75. Also for 1, which they call it clear match, is high. So that's it. You can continue seeing it. HLA matching in renal transplantation is possible because donor kidney can be stored for some time before being transplanted, and patient needing a kidney allograft can be maintained on dialysis until a well-matched organ is available. In case of heart and liver transplantation, organ preservation is more difficult and potential recipients are often in critical condition. For this reason, HLA typing is not considered in pairing of potential donor and recipients, and the choice of donor and recipients is based on ABO group matching. Other measures of immunologic compatibility described later, and anatomical compatibility. Deposit of heart donor, the emergent need for transplantation, and the success of immunosuppression override any benefit of reducing HLA mismatch between donor and recipients. As we will discuss later, in HSC, hematopoietic stem cells transplantations, HLA matching is essential to reduce risk for graft versus host disease. Most of HLA haplotype determination are now performed by polymerase chain reaction (PCR), replacing older theological methods. MHC genes can be amplified by PCR with the use of primer that bind to null-polymorphic sequence within 5 to 3 primer end of exons, encoding the polymorphic region of class 1 and class 2 MHC molecules. The amplified segment of DNA can then be sequenced. Thus, the actual nucleotide sequence and therefore the predicted amino acid sequence can be directly determined for MHC alleles of any cells, providing a precious molecule's tissue typing. On the basis of this DNA sequencing effort, the nomenclature of HLA alleles has changed to reflect the identification of many alleles not distinguished by previous serological method. Each allele defined by sequence has at least a 4-digit number, but some alleles require 6 or 8 digits for precise definition. The first 2 digits usually correspond to the older serological defined allotypes, and the third and fourth digit indicate subtypes. Allele with the difference in the first 4 digits encode protein with different amino acid. For example, HLA DRB1 star 1301 is the sequence defined 01 allele of serological defined HLA-DR13 family of gene encoding HLA-DR beta 1 proteins. Patients in need of allograft are also tested for the presence of preformed antibody against the donor MHC molecules or other cell surface antigen Two types of tests are done to detect this antibody. In the panel reactive antibodies test, patients waiting for the organ transplant are screened for the presence of preformed antibody reactive to allogenic HLA molecules prevalent in population. The presence of this antibody, which may be produced as a result of previous pregnancy, transfusion, transplantation, increases the risk of hyperacute or acute vesicular rejection. Small amount of the patient's serums are mixed with multiple fluorescently labeled beads coated with defined MSC molecules representing representative of MSC alleles that may be present in an organ donor population. Each MSC molecule is attached to the bead with a different colored fluorescence label. Binding of the patient's antibody to beads is determined by flow cytometry. The results are reported as percentage of the MSC allele panel with which the patient's serum reacts. The PRA is determined on multiple occasions while patients wait for organs or allografts. This is because the PRA vary as each panel is chosen at random and patient serum antibody titer may change over the time. If potential donor is identified, the cross-matching test will determine if the patient has antibody that reacts specifically with that donor cells. The test is performed by mixing the recipient's serum with donor blood lymphocytes, a convenient source for cells, some of which express both class 1 and class 2 MHC proteins. Complement-mediated cytotoxic testing or flow cytometric assays can then be used to determine if the antibodies and the recipient serums have bound to the donor cells. For example, complement is added to a mixture of cells and serums, and if preformed antibody, usually against donor MSC molecules, are present in recipient serums, the donor cells are lysed. This will be a positive cross-match, which indicates that the donor is not suitable for the recipients. So those are things that you will see. Mechanism of active immunosuppressive drugs. Each major category of drug used to prevent and treat allograft rejection is shown along with the molecular targets of the drug. So now we are going to discuss the drugs and see how they work. Immunosuppression to prevent or to treat allograft rejection. Immunosuppressive drugs that inhibit or kill T lymphocytes are principal agents used to treat or prevent graft rejection. Several drugs are commonly used. Inhibitors of T cell signaling pathway. The calcitonurins inhibitor, cyclosporin and tacrolimus, inhibit transcription of certain genes in T cells, most notably genes encoding cytokines such as interleukin-2. Cyclosporin is the fungal peptide that binds with high affinity to a ubiquitous cellular protein called cyclophilin. The complex of cyclosporin and cyclophilin bind to and inhibit the enzymic activity of calcinurin. Calcinurin or calmodin activated serine threonine phosphatase, we have seen already in chapter 7. Because calcinurin is required to activate transcription factor, nuclear factor activated T cells, and cyclosporin inhibit NF and the nuclear factor activated T cells activation and the transcription of interleukin-2 and other cytokine genes. The net results is that cyclosporin blocks the interleukin-2 dependent proliferation and differentiation of T cells. Tacrolimus is a macrolide made by bacterium that function like cyclosporin. Tacrolimus bind to FK506 binding proteins and the complex share with the cyclosporins complex the ability to bind calcinurin and inhibit these activities. The introduction of cyclosporin into clinical practice ushered in the modern era of transplantation. Before the use of cyclosporin, the majority of transplanted heart and liver were rejected. Now, as a result, the use of cyclosporin, tacrolimus, and other more recently introduced drugs, the majority of these allograft survive for more than 5 years. Nevertheless, calcinurins inhibitors have limitations. For example, at a dose needed for optimal immunosuppression, cyclosporin causes kidney damage. and some rejection episodes are reflected to cyclosporin treatment. Tacrolimus was initially used for liver transplant recipients, but it has now largely replaced cyclosporin for all organ transplant because of better efficacy and safety. The immunosuppressive drug rampamycin, sometimes called sirolimus, inhibit growth factor-mediated T cell proliferation. Like tacrolimus, rapamycin bind to FKBP, but the rapamycin FKBP complex does not inhibit calcineurins. Instead, this complex binds to and inhibits a cellular enzyme called mTOR, mechanistic target of rapamycin, which is the serine threonine protein kinase required for translocation of protein that promote the survival and proliferation. mTOR is negatively regulated by proteins complex called tuberous sclerosis complex 1s. The phosphatidylinosyl 3 kinase PI3K AKT signaling result in phosphorylation of TSC2 and release the mTOR inhibition. Several growth factor receptor signaling pathway, including the interleukin-2 receptor pathway in T cells, as well as T cell receptor and the CD28 signals, activate the mTOR through the PI3K-AKT, leading to translocations of translation of proteins needed for cell cycle progression. Thus, by inhibiting mTOR function, rapamycin block T cell proliferation. Combination of calcineurin inhibitor, which block interleukin-2 synthesis, and rapamycin, which block interleukin-2 driven proliferation, potently inhibit T cell response. Interestingly, rapamycin inhibits the generation of effector T cell but does not impair the survival and function of regulatory T cells, as such, which may promote immune suppression of allograft rejection. mTOR is involved in dendritic cell function, and therefore rapamycin may suppress T cell response by its effects on B cells as well. mTOR is also involved in B cells proliferation and antibody response, and therefore rapamycin also may be effective in preventing or treating antibody-mediated rejection. Other molecule involved in cytokine and T cell receptor signal are also target of immunosuppressive drugs that are in trial for treatment and prevention of allographic rejection. These target molecules include tyrosine kinase JAK3, which is involved in signaling by various cytokine receptor including interleukin-2, and protein kinase C, an essential kinase in T cell receptor signaling. Antimetabolites. Metabolic toxin that kills proliferative T cells are used in combination with other drugs to treat graft rejection. These antigen inhibit proliferation of lymphocyte precursor during their maturation and also kill proliferating mature T cells that have been stimulated by alloantigens. The first such drugs to be developed for prevention and treatment of rejection was azathioprine. This drug is still used, but it is toxic to precursor of leukocytes in the bone marrow and enterocytes in the gut. The most widely used drug in this class is mycophenolate mofetil. Mycophenolate mofetil is metabolized to mycophenol oxide, which block the activities of ionosines monophosphate dehydrogenase, an enzyme required for de novo synthesis of guanine nucleotides. Because proliferating lymphocytes are particularly dependent on de novo synthesis of protein, MMF target lymphocyte in a relatively specific manner. MMF is now routinely used often in combination with tacrolimus to prevent acute allograft rejections. Function blocking and depleting anti-lymphocyte antibodies. Antibodies that react with T cells surface structure and deplete or inhibit T cells are used to treat acute rejection episode. The first T cells antibody used in transplantation was a mouse monoclonal antibody called OCT3 that is specific for human CD3. OCT3 was the first monoclonal antibody used as drug in human, but it is no longer being produced. Polyclonal rabbit of horse antibody specific for a mixture of human T cell surface protein, so-called antithymocytes globulin, also have been in clinical use for many years to treat acute allograft rejection. These anti-T antibodies deplete circulating T cells either by activating the complement system to eliminate T cell or by opsonizing them for phagocytosis. Monoclonal antibodies specific for CD25, the alpha subunit of interleukin 2 receptor, are now in clinical use. These reagents prevent T cell activation by blocking interleukin 2 binding to activate T cell and interleukin 2 signaling. Another monoclonal antibody in use is in clinical transplantation is one specific for CD52, a cell surface protein expressed widely on mostly mature B and T cell whose function is not understood. Antibody CD52 called alemtuzumab was originally developed to treat B cell tumor, and it was found to be profoundly deplete most peripheral B cells and T cells for many weeks after injection into patients. In the initial clinical trial, it was administered just before and after transplantation, with hope that it may induce prolonged state of graft tolerance, but it's not widely used now. The major limitation to the use of monoclonal polyclonal antibodies from other species that human given these agents produce anti-Ig antibody that neutralize the injected foreign Ig. For this reason, humanized antibodies Ig against CD3 and CD25, which are less immunogenic, have been developed. Costimulatory blockage. Drugs that block T cell costimulatory pathway reduce acute allographic rejection. The rationale for use of these types of drugs is to prevent delivery of costimulatory signal required for activation of T cells. Recall that CTLA-4 is a recombinant protein composed of extracellular portion of CTLA-4 fused to an IgG-Fc domain. A high affinity forms of CTLA-Ig called belataceps, which bind to B7 molecules on antigen-presenting cell, prevent them from interacting with T cells. CD28 is approved for use in allograft recipients. Clinical studies have shown that belatacept can be as effective as cyclosporine in preventing acute rejection, but its high cost and other factors have limited widespread use for these biological agents. An antibody that binds to T cell CD40 ligands and prevent its interaction with CD40 and antigen-presenting cells has proved beneficial for preventing graft rejection in experimental animals. In some experimental protocol, simultaneously blockade of both B7 and B40 appear to be more effective than either alone, preventing graft to survive. However, in clinical trial of anti-CD40 ligands antibody, patients developed thrombotic complication apparently related to the expression of CD40 ligands on human platelets. Treatment to reduce alloantibodies and alloreactive B cells. As we have learned more about importance of alloantibodies in mediating acute and perhaps chronic rejection, therapies targeting the antibodies and B cells that were developed for other diseases are now being used in transplant patients. For example, plasmapheresis is a method used to treat acute antibody-mediated rejection. In this procedure, a patient's blood is pumped through a machine that removes the plasma but return the blood cells to circulation. In this in this way, circulating pathogenic alloreactive antibody can be removed. Intravenously, immunoglobulin IVIG, which is an accepted therapy for several antibody-mediated inflammatory disease, is also used to treat acute antibody-mediated rejection. In IVIG therapy, pooled IgG from normal donor is injected intravenously into patients. The mechanism of action are not fully understood, but likely involves binding of injected IgG to patient Fc receptor on various cell types, thereby reducing alloantigens production and blocking effector function of the patient's own antibodies. IVIG also enhances the aggregation of the patient's antibody by competitively inhibiting their binding to the neonatal Fc receptor. B-cell depletion by administration of rituximab, an anti-20 antibody that is approved for treatment of B-cell lymphoma and autoimmune disease, is used in some cases for acute antibody-mediated rejection. The proteasome inhibitor botensilimab, which kill plasma cells and is approved to treat multiple myeloma, is also sometimes used to treat antibody-mediated allograft rejection. Anti-inflammatory drugs. Anti-inflammatory agents, specifically corticosteroids, are frequently used to reduce inflammatory reaction to organ allograft. The proposed mechanism of action of these natural hormones and their synthetic analog is to block the synthesis and secretion of cytokines, including tumor necrosis factor and interleukin-1s, and other inflammatory mediators such as prostaglandin, reactive oxygen species, and nitric oxide, produced by macrophage and other inflammatory cells. The net result of this therapy is reduced leukocyte recruitment, inflammations, and graft chain damage. Current immunosuppressive protocols have dramatically improved graft survival. Before the use of calcineurins inhibitor, the 1-year survival rate of unrelated cadaver kidney graft was between 50 to 60, with 90% rate of graft from living related donor, which are better matched in the recipients. Since cyclosporins, tacrolimus, rapamycin, and MMF have been introduced, the survival rate of unrelated cadaveric kidney graft have increased approximately 90 at first year. Heart transplantation, for which HLA matching is not practical, has also significantly benefited from the use of various classes of immunosuppressive drugs, reviewing earlier, and now has similar approximately 90% 1-year survival rate and approximately 75% 5-year survival rate. Experience with other organs is more limited, but the survival rate has improved with modern immunosuppressive therapies, with 10-year patient survival rates approximately 60 to 75% for the pancreas and liver recipients, respectively, and 3 years patient survival rate of 70 to 80 for lung recipients. Strong immunosuppression is usually started in allograft recipients at the time of transplantation with combination of drug called induction therapy. After a few days, the drugs are changed to long-term maintenance of immunosuppression. For example, in the case of adult kidney transplantation, a patient may be initially induced with an anti-interleukin-2 receptor or anti-T cell depleting antibody and a high-dose corticosteroid, and then maintained on calcineurins inhibitor and antimetabolites and maybe low dose of corticosteroid. Acute rejection, when it occurs, it is managed by rapidly intensifying the immunosuppressive therapy. In modern transplantation, chronic rejection has become more common cause of allograft failure, especially in cardiac transplantation. Chronic rejection is much less response to immunosuppressions than in acute rejection. Immunosuppressive therapies lead to the increased susceptibility to the infection and tumor. The major goals of immunosuppression is to treat graft rejection. The major goals of immunosuppression is to treat graft rejection. The major goals of immunosuppression to treat graft rejection is to reduce generation and function of helper T cells and cytotoxic T lymphocyte, which mediate acute cellular rejection. It is therefore not surprising that defense against viruses and other intracellular pathogens and tumor, the physiological function of T cell, is also compromised in immunosuppressive transplant recipients. A frequent problem in immunosuppressive patients is reactivation of latent hyperviruses, including cytomegalovirus, herpes simplex viruses, varicella zoster viruses, Epstein-Barr viruses. For this reason, transplant recipients are now given prophylactic antiviral therapy for herpes viruses infection. Immunosuppressive allograft recipients are also at great risk of the variety of opportunistic infection, which normally do not occur in immunocompetent people, including fungal infection, pneumocystis jirovecii, pneumonia, histoplasmosis, coccidiomycosis, and microsporidiosis, and protozoa infection like toxoplasmosis and cryptosporidiosis. Immunosuppressive allograft recipients have higher risk of developing the cancer compared with general population, includes various forms of skin cancer. Some of the tumors that are more frequently found in allograft recipients are known to be caused by viruses, and therefore they may arise because of impaired antiviral immunity. These include uterine cervical carcinoma, which is related to human papilloma virus infection, lymphoma caused by Epstein-Barr virus infection. The lymphoma found in allograft recipients are called the post-transplantation lymphoproliferative disorder, and most are derived from the Epstein-Barr virus infected B lymphocyte. Despite the risk for infection and neoplasm associated with the use of immunosuppressive drugs, the major limitation of the most of these drugs, including calcineurin inhibitor, mTOR inhibitor, antimetabolites, and corticosteroid, is directly toxic to cell and related to immunosuppression. In some cases, the toxicity affects the cells as rejection does, such as cyclosporin toxic to renal tubular epithelial cells, which can complicate the interpretation of declining renal function in kidney allograft recipients. Method to induce donor-specific tolerance. Allograft rejection may be prevented by making the host tolerance to alloantigen of the graft. Tolerance in this setting means that host immune system does not injure the graft despite the withdrawal of immunosuppressive agents. It is presumed that tolerance to allograft will involve the same mechanism that involved in tolerance of self-antigens, of seen in chapter 15. That is, energy deletion and active suppression of allograft T cells by Treg. Tolerance is desirable in transplantation because it could be allogenic specific and will therefore avoid the major problem associated with non-specific immunosuppressions, namely immunodeficient, leading to increasing susceptibility to infection and development of tumor and drug toxicity. In addition, achieving graft tolerance may reduce chronic rejection, which has to date been unaffected by the commonly used immunosuppressive agents that prevent and reverse acute rejection episode. Various experimental approach and clinical observation have shown that it should be possible to achieve tolerance to allograft. In experiment in mice, Medwar and colleague found that if neonatal mice of one strain, the recipients, are given spleen cells of another strain, the donor, the recipient will subsequently accept skin graft from the donor. Such tolerance is in allogenic specific because recipient will reject graft from mouth strain that expresses MHC allele that differ from those of spleen cells donor. Renal transplant patients who have received blood transfusion containing allogenic leukocyte have lower incidence of acute rejection episode than do those who have not been transfused. The postulated explanation for this effect is that the introduction of allogenic leukocyte by transfusion produces tolerance to alloantigens. One underlying mechanism for tolerance induction may be that transfused donor cells contain immature dendritic cells, which induce unhyporesponsiveness to donor alloantigens. Indeed, pretreatment of the potential recipients with blood transfusion is now used as prophylactic therapy to reduce rejection. Several strategies are being tested to induce donor-specific tolerance in allograft recipients. Costimulatory blockade. It was postulated that recognition of alloantigens in the absence of costimulation will lead to the T cell tolerance, and there is some experimental evidence in animal to support this. However, the clinical experience with agents that block costimulation is that they suppress immune response to allograft but not to induce long-lived tolerance, and the patient have to be maintained on therapy. Hematopoietic chimerism. We mentioned earlier that transfusion of donor blood cells into the grafted recipients inhibit rejection. If the transfusion donor cells or progenies of the cells survive for extended period in the recipients become chimera. Long-term allographic tolerance by hematopoietic chimerism has been achieved in small number of renal allographic recipients who received a hematopoietic stem cells transplant from the donor at the same time as organs are grafted. But the risk of uh, hematopoietic stem cell transplantation and the availability of appropriate donor may limit the applicability of this approach. Transfer of induction of Treg. Attempt to generate donor-specific Treg in culture and transfer these into graft recipients are ongoing. There have been some success reported in recipients of hematopoietic stem cell transplant in whom infusion of Treg reduced the GVHD. In rare cases, some transplant survival and function even after immunosuppression is stopped or significantly reduced because of infections or toxicity. This has been most frequently observed in liver transplant patients. Clinicians use the term operational tolerance to refer to this phenomenon. It is not clear in most cases if alloreactive T cells response are reduced or extinguished. It's also not known by why this occurs most often with liver transplantation. Xenogenic transplantation. The use of solid organ transplantation as clinical therapy is greatly limited by inadequate number of donor organs available. For this reason, the possibility for transplantation of organs from mammals such as pig into human recipients has kindled great interest. A major immunological barrier to xenogenic transplantation is the presence of natural antibody in human recipients that cause hyperacute rejection. A major immunological barrier to xenogenic transplantation is the presence of natural antibodies in human recipients that causes hyperacute rejection. More than 95% of the primates have natural IgM antibodies that are reactive with the carbohydrate determinants expressed by cells of the species that are evolutionarily distant, such as pig, which have organs that are anatomically compatible with humans. The majority of human anti-pig natural antibodies are directed at carbohydrate determinant formed by the action of the pig alpha-galactose transferase enzymes. This enzyme places an alpha-linked galactose moiety on the same substrate that in the human and other primate cell is fucosylized to form the blood group H antigen. Investigator has produced alpha-galactose transferase genes knockout pig to try to circumvent this problem, but this strategy alone has not been successful. Humans rarely produce natural antibody against carbohydrate determinant of closely related species such as chimpanzee. Thus, organs from chimpanzee or other higher primate might theoretically be accepted in human. However, ethical and logistic concerns have limited such a procedure. Natural antibody against xenograftic induces hyperacute rejection by the same mechanism as those seen in hyperacute allograftic rejection. These mechanisms include the generation of endothelial cell procoagulants, and platelet aggregation substance, coupled with the loss of endothelial anticoagulants mechanism. However, the consequence of activation of human complement on pig cells are typically more severe than the consequence of activation of complement by natural antibody on human allogenic cells. This may be because some of the complement regulator protein made by pig cells are not able to interact with human complement protein, and thus can limit the extent of injury induced by human complement system. For this reason, investigators have developed genetically modified pig that are transgenic for human complement regulatory proteins. The CRISPR-Cas9 genes editing technology is now being applied to generate pig with multiple genetic modifications chosen to reduce xenographic rejection. Even when hyperacute rejection is prevented, xenografts are often damaged by a form of acute vesicular rejection that occur within 2 to 3 days of transplantation. This form of rejection has been called delayed xenographic rejection, accelerated acute rejection, or acute vesicular rejection, and is characterized by intravesicular thrombosis and of necrosis of vessel walls. The mechanism of delayed xenograftic rejections are incompletely understood. Recent findings indicate that may be incompatibility between primate platelets and porcine endothelial cells that promote thrombosis independent of antibody-mediated damage. Xenograft also can be rejected by T cell mediated immune response to xenoantigens. The mechanism of cell mediated rejection of xenograft are thought to be similar to those that we have described for allograft rejection. Blood transfusion and the ABO and RH blood group antigen. Blood transfusion is a form of transplantation in which whole blood or blood cells from one or more individuals are transferred intravenously into the circulation of another individual. Blood transfusion are the most often performed to replace blood lost for hemorrhage or to correct defect caused by inadequate production of a blood cell, which may occur in a variety of diseases. The major barrier to successful blood transfusion is the immune response to cell surface molecules that differ among individuals. The most important alloantigen system in blood transfusion is the ABO system, which we'll discuss in detail later. Individuals who do not express a particular blood group's antigens produce a natural IgM antibody against that antigen. If such individuals are given blood cells expressing that antigen, the pre-existing antibody bind to transfuse the cells, activate complement, and cause transfusion reaction, which can be life-threatening. Transfusion across an ABO barrier may trigger an immediate hemolytic reaction, resulting in both intravascular lysis and red blood cells of the red blood cells, and probably mediated by complement system and extensive phagocytosis of antibody and a complement-coated erythrocytes by macrophage in the liver and spleen. Hemoglobin is liberated from the lyzed red blood cells in a quantity that may be toxic for kidney cells, causing acute renal tubular cells necrosis and kidney failure. High fever, shock, and disseminated intravesicular coagulation may also develop, suggestive of release of massive amount of cytokine, tumor necrosis factor, or interleukin 1. The disseminated intravesicular coagulation consumes clotting factor faster than they can be synthesized, and the patient may paradoxically die of bleeding in the presence of widespread clotting. More delayed hemolytic reaction may result from incompatibility of minor blood group antigen. This results in progressive loss of transfused red blood cells, leading to anemia and jaundice, and later a consequence of overloading the liver with hemoglobin-derived pigments. ABO blood group antigens. The ABO antigen are carbohydrates linked to cell surface proteins and lipids, which are synthesized by polymorphic glycotransferase enzymes that vary in activity depending on the inherited allele. The ABO antigen were first alloantigen system to be defined in mammal. The specific carbohydrate antigen are present on red blood cells, hence their name but they are also found on the endothelial and some epithelial cells. All normal individuals produce a common core glycan, which is attached mainly in the plasma membrane proteins. Most individuals possess a focal cell transferase that add the fucose moiety to a non-terminal sugar residue for core glycan, and the fucosylated glycan is called H antigen. A single gene on chromosome 9 encodes a glycosyltransferase enzyme that may further modify the H antigen. There are three allelic variants of these genes. The O allele gene's product is devoid of enzymic activities. The A allele encoded enzymes transfer the terminal any acetyl galactosamine moiety onto H antigens, and B allele genes product transfer a terminal galactose moiety. Individuals who are homologous for O allele cannot attach terminal sugar to H antigens and expresses only H antigen. In contrast, individuals who possess an A allele, AA homozygous or AO heterozygous or AB heterozygous form the A antigen by adding a terminal any acetylgalactosamine to some of their H antigen. Similarly, individual who expresses B allele, BB homozygous, BO heterozygous, or AB heterozygous form a B antigen by adding a terminal galactose to some of their H antigen. AB heterozygous form both A and B antigen from some of their H antigens. The terminology has been simplified so that AOO individuals are said to be blood group O, AA and AO individual are blood group A, BB and BO individual are blood group B, and AB individual are blood type AB. Mutation in these genes encoding the fucosyl that produce the H antigen are rare. People who are homozygous for such a mutation are said to have Bombay blood groups and cannot produce HA or B antigen. Individual who expresses a particular A or B group's antigen are tolerant to that antigen, but individuals who do not express that antigens produce natural antibodies that recognize the antigen. Almost all individuals express the H antigen and therefore are tolerant to this antigen and do not produce anti-H antibodies. Individuals who express A or B antigens are tolerant to these molecules and do not produce anti-B or anti-B, anti-A or anti-B antibodies respectively. However, blood group O and A individual produce anti-B IgM antibodies, and blood group O and B individual produce anti-A IgM antibodies. Individuals with the Bombay phenotype who are unable to produce the core H antigen make antibody against H, A, and B antigens. On face value, it seems paradoxical that individuals who do not express blood group antigen make antibody against it. The likely explanation is that antibodies are produced against glycolipid of intestinal bacteria that happen to cross-react with ABO antigens unless the individual is tolerant to one or more of these. Predictably, the presence of any blood group antigen on an individual's red blood cell induces tolerance to that individual. So this is the diagram that you can see, and all the explanation have been said. In clinical transfusion, the choice of blood donor of particular recipients is based on the expression of blood group antigens and antibody response to them. If patients receive a transfusion of red blood cells from a donor who expresses the antigen not expressed on self red blood cell, a transfusion reaction may result described earlier. It followed that AB individual can tolerate transfusion from all potential donor and are therefore called universal recipients. Similarly, O individual can tolerate transfusion only from O donor, but can provide blood to all recipients and therefore called universal donor. In general, difference in minor blood groups lead to red blood cell lysis only after repeated transfusion trigger a secondary antibody response. A and B blood group antigen are expressed on many other cell types in addition to blood cells, including endothelial cell. For this reason, ABO typing is critical to avoid hyperacute rejection and certain solid organ allografts, as discussed earlier in the chapter. ABO incompatibility between mothers and fetus generally does not cause problem for the fetus because most anti-carbohydrate antibodies are IgM and cannot cross the placenta. Other blood group antigens. Lewis antigen, the same glycoprotein that carry the A and B blood group determinant, can be modified by other glycosyltransferase to generate a minor blood group antigen. For example, addition of fructose moiety at other non-terminal position can be catalyzed by different fructose transferases and create epitope of the Lewis antigen system. Lewis antigen have received much attention from immunologists because these carbohydrate groups serve as ligand for E-selectin and P-selectin and thus play a role in leukocyte migrations. They are not, however, involved in strong transfusion reaction because individuals do not make a natural antibody against them. Oh, good. Rhesus antigen. The rhesus RH antigen, named after a monkey species in which they were originally identified, are another clinically important set of blood group antigens. A rhesus antigen are non-glycosylated hydrophobic cell surface protein found in red blood cells membrane and are structurally related to other red blood cells membrane protein with the transporter functions. Rhesus proteins are encoded by two tightly linked and highly homologous genes, but only one of them, called RHD, is commonly considered in clinical blood typing. This is because up to 15% of population has a deletions or other alteration of RHD allele. These people, called RHA negative, are only tolerant to RHD antigen and will make antibodies to antigens if they are exposed to Rh positive cells. The major clinical significance of anti-Rh antibodies is related to the hemolytic reaction in developing fetus that are similar to transfusion reaction. Rh negative mother carrying Rh positive fetus can be sensitized by fetal red blood cells that enter the maternal circulations, usually during childbirth. Because of Rh antigen is proteins as opposed to the carbohydrate ABO antigens, class switching high affinity IgG antibody are specific for Rh are produced in Rh negative mother. Subsequent pregnancy in which the fetus is Rh positive are at risk because the maternal and Rh IgG antibodies can cross the placenta and mediate the destruction of the fetal red blood cell. This causes hemolytic disease of the fetus and newborns, also called erythroblastosis fetalis, and can be lethal for the fetus. This disease can be prevented by administration of anti-RHD antibodies for the mother within 72 hours of the birth of the first Rh positive baby. The treatment prevents the baby's Rh positive red blood cells that enter the mother's circulation from inducing the production of anti-Rh antibodies in the mother. The exact mechanism of action by administration of antibody are not clearly, but may include phagocytic clearance of or complement-mediated lysis of the baby red blood cells before they can elicit an antibody response in the mother, or Fc receptor dependent feedback inhibition of the mother RHD specific B cells. Hematopoietic stem cells, HSC transplantation. The hematopoietic stem cell transplantation is clinical procedure to treat diseases caused by intrinsic defect in one or more hematopoietic lineage, and more commonly to treat cancer of the blood cells. In this procedure, the patient's own hematopoietic cells are destroyed and the hematopoietic cells from the healthy donor are then given to patients. We consider a hematopoietic stem cell transplantation separately from other form of transplantation because this type of grafting has several unique features that are not encountered with solely organ transplantation. Indications, method, and immune barrier in hematopoietic stem cell transplantation. The transplantation of pluripotent hematopoietic stem cell was done in past using an inoculum of the bone marrow cells collected by aspiration, and the procedure is often called bone marrow transplantation. In modern clinical practice, hematopoietic stem cells are more often obtained from the blood of donor after treatment of donor with colony stimulating factor that mobilizes stem cells from the bone marrow. The recipient is treated before transplantation with the combination of chemotherapy, immunotherapy, and irradiation to free up nick of transferred stem cells, and in case of treating hematopoietic stem malignancy, to kill as many as cancer cells as possible. After transplantation, the injected stem cells repopulate the recipient's bone marrow and differentiate into all hematopoietic stem lineage. Hematopoietic stem cells transplantation is the most often used clinically in the treatment of leukemias and pre-leukemic condition. The mechanism by which hematopoietic stem cell transplantation cure hematopoietic stem neoplasm is in part of the graft versus tumor effect, in which mature T cell present in the bone marrow or stem cell inoculum recognize alloantigen on residue tumor cells and destroy them. Natural killer cells is injected. A natural killer cell in the injected hematopoietic stem cell inoculum may also recognize and kill leukemic cells. Autologous hematopoietic stem cells transplantation is used to treat plasma cell derived tumor called myeloma. Because many of the neoplastic cells are in the patient bone marrow, these are killed when the bone marrow is ablated as part of conditioned regimes. The hematopoietic stem cells transplanted are from the same patient, but may, they do not contain myeloma cells because these cells do not circulate in the blood. Thus, the function of bone marrow is restored and tumor cells are eliminated. Since the transplant is autologous, there is no risk for its rejection, described later. Hematopoietic stem cell transplantation is also used clinically to treat disease caused by inherited mutation in gene affected only cell derived from hematopoietic stem cells, such as lymphocyte or red blood cell. Example of such disease that can be treated by hematopoietic stem cell transplantation are X-linked severe immunodeficiency diseases, SCID, IPEX, the disease caused by FOX P mutation and deficiency of regulatory T cells, and hemoglobin mutations such as beta thalassemia major and sickle cell disease. XKID has also been treated by expressing the normal genes in patients' own hematopoietic stem cell and transplanting them correct the deficiency. The same approach is applicable to other diseases in which the defective gene is known and is functional in blood cell which are produced from a hematopoietic stem cell. Because these transplants are autologous, they do not cause immunological problems. Allogenic hematopoietic stem cells are rejected by even minimal incompetent hosts, and therefore the donor and recipient must carefully be matched at all MHC log. The mechanism of rejection of hematopoietic stem cell are not completely known, but in addition to adaptive immune mechanism, hematopoietic stem cell may be rejected by natural killer cell. The role of natural killer cells in bone marrow rejection has been studied in experimental animal. Irradiated F1 hybrid mice reject bone marrow cells donated by either inbred parents. This phenomenon, called hybrid resistance, appear to violate the classical law of solid organ transplantation, in which F1 mice do not react against the grafted from either parents. C. Hybrid resistance is seen in T cells deficient mice and depletion of recipient natural killer cell in anti-natural killer cell antibody prevent rejection of parental bone marrow cells. Hybrid resistance is probably due to the host natural killer cell reacting against bone marrow precursor that lack the class 1 MHC molecule expressed by host. Recall that normally recognition of self class 1 MHC inhibits the activation of natural killer cells, and if these self MHC molecules are missing, the natural killer cells released from inhibitions. Even after successful engraftment, two additional problems are frequently associated with the hematopoietic stem cell transplantation, that is, GVHD and immunodeficiency, which are discussed next. Immunological complication of hematopoietic stem cell transplantation. Graft versus host disease. Grafted versus host disease is caused by reaction of grafted mature T cells in hematopoietic stem cell inoculation with allergens of host. It occurs when the host is immunocompromised and therefore unable to reject the allogenic cells in the graft. In most cases, the reaction is directed against the minor histocompatibility antigen of the host because bone marrow transplantation is not usually performed when the donor and the recipient have difference in MHC molecules. Graft versus host disease may also develop when solid organs that contain a significant number of T cells are transplanted, such as small bowel or a small bowel, lung, or liver. Graft versus host disease is the major limitation to success of bone marrow transplantation. Immediately after graft versus immediately after hematopoietic stem cell transplantation, immunosuppressive agents including calcineurin inhibitor, cyclosporin, and tacrolimus antimetabolites such as methotrexate and mTOR inhibitors, sirolimus, are given for prophylactic against developing the grafted versus host diseases. Despite this aggressive prophylactic strategy, graft versus host disease is the principal cause of mortality among hematopoietic stem cell transplant recipients. Graft versus host disease may be classified on the basis of histologic pattern into acute and chronic forms. Acute graft versus host disease is characterized by epithelial cell death in the skin, liver, mainly biliary epithelial, gastrointestinal tract, and it is manifested clinically by rash, jaundice, diarrhea, and gastrointestinal hemorrhage. When epithelial cell death is extensive, the skin and lining of the gut may slough off. In circumstance in this circumstance, acute graft versus host disease may be fatal. Chronic graft versus host disease is characterized by fibrosis and atrophy of one or more of the same organ without evidence of acute cell death. The chronic graft versus host disease may also involve the lungs and produce obliterations of small airway called bronchiectasis obliterans, similar to what is seen in chronic rejection of lung allografts. When this is severe, chronic GVHD leads to the complete dysfunction of affected organs. In animal model, acute GVHD is initiated by mature T cell transferred with hematopoietic stem cell and elimination of mature donor T cells from the graft can prevent development of GVHD. In clinical, hematopoietic stem cell transplantation efforts to eliminate T cells from inoculum have reduced the incidence of GVHD, but also decreased the graft versus leukemia effects that is often critical in treating leukemias by this type of transplantation. T-cell depleted hematopoietic stem cell preparation also tend to engraft poorly, perhaps because mature T-cell produce colony-stimulating factor that aid in stem cell repopulation. Although GVHD is initiated by grafted T-cells recognizing host antigens, the effector cells that cause epithelial cell injury are less well defined. On histological examination, natural killer cells are often attached to dying epithelial cells, suggesting that natural killer cells are important effector cells of acute GVHD. CD8 T cells, cytotoxic T cells, and cytokines also appear to be involved in tissue injury in acute GVHD. You will see the diagram, histological diagram. The relationship of chronic GVHD to acute GHD is not known and raises issues similar to those of relating chronic allographic rejection to acute allographic rejection. For example, a chronic GVHD may represent the fibrosis of the wound healing secondary to acute loss of epithelial cell. However, chronic GVHD can arise without evidence of prior acute GVHD. An alternative explanation is that chronic GHVD represent a response to ischemia caused by vascular injury. Both acute and chronic GVHD are commonly treated with intense immunosuppression, such as high dose of corticosteroid, but many patients do not respond well. Therapeutic failure may be because of treatment target only some of the many effector mechanisms play in GVHD, and some treatment may deplete Treg, which are important for preventing GVHD. The BTK, Bruton tyrosine kinase inhibitor, ibrutinib, which had been previously approved for treating B-cell malignancies, also has been shown to in clinical trial to be effective in treating the chronic GVHD and has been approved by the US Food and Drug Administration for this purpose. Therapies in development include Treg cell transfer. Immunodeficiency after hematopoietic stem cell transplantation. Hematopoietic stem cell transplantation is often accompanied by immunodeficiency. Several factors may contribute to the defective immune response in recipients. The transplant recipients may be unable to generate a complete new lymphocyte repertoire. Radiation therapy and chemotherapy used to prepare recipients for transplantation may deplete patient memory cells and long-lived plasma cells, and it can take long time to regenerate these populations. The consequence of immunodeficiency is that hematopoietic stem cell transplant recipients are susceptible to viral infection, especially reactivation of cytomegalovirus infection, and to many bacteria and fungal infections. They are also susceptible to Epstein-Barr virus-provoked B lymphomas. The immune deficiency of hematopoietic stem cell transplant recipients can be more severe than those of conventionally immunosuppressed patients. Therefore, the recipients commonly receive prophylactic antibiotic, antiviral prophylaxis to prevent cytomegalovirus infection, antifungal prophylaxis to prevent invasive aspergillosis infection. Recipients are also immunized against common infection to restore the protective immune that is lost because of ablation of bone marrow before hematopoietic stem cell transplantation. There is a great interest in the use of pluripotent stem cells to repair tissue that have little natural regenerative capacity, such as cardiac muscle, brain, and spinal cord. One approach is to use embryonic stem cells, which are pluripotent stem cells derived from the blastocyst stage of human embryo. Although embryonic stem cells have not yet been widely used clinically, it's likely that a major barrier to their successful grafting will be their alloantigenicity and rejection by the recipient's immune system. A possible solution to this may be used induced proton stem cell, which can be derived from adult somatic tissue by transduction of certain genes. Immuno, immuno, Immunologic advantage of the induced pluripotent stem cells approach is that these cells can be derived from somatic cells harvested from the patients, and therefore they will not be rejected. However, the current experience is that induced pluripotent stem cells do not efficiently generate mature, stable population in vivo, so their potential for cell replacement may be limited. Another solution being investigated is to remove major histocompatibility genes from allogenic embryonic stem cell by CRISPR-Cas9 genome editing technology. Summary: allograftic are tissue or organ transplanted from one individual to a genetically non-identical recipients. Allografts stimulate a specific immune response called rejection that can destroy the graft. The major molecules targets in allograft rejection are allogenic class 1 and class 2 major histocompatibility complex MHC molecule. Intact allogenic MHC molecule may be presented on the donor antigen presenting cells to recipient T cell direct recognition, or allogenic MSC molecule may be internalized by host antigen presenting cell that enter the graft, reside in draining lymphoid organs, and be processed to present and presented to T cell as peptide associated with self MSC molecule in direct recognition. The frequency of T cell capable of recognize allogenic MSC molecule is very high compared with T cells that recognize any microbial peptide bound to self MSC, explaining why the response of alloantigen is much stronger than response to conventional foreign antigen. Graft rejection is mediated by T cell, including cytotoxic T cell lymphocyte that kill the graft cells, and helper T cell that cause cytokines mediated inflammations resembling delayed type hypersensitivity reaction, and by antibodies. Several effector mechanisms cause rejection of solid organ graft. Pre-existing antibodies specific for donor blood groups, MHC, and other antigen cause hyperacute rejection characterized by thrombosis of the graft vessels. Autoreactive T cells and antibody produced in response to the graft causes blood vessel wall damage and parenchyma cell death, called acute rejection. Chronic rejection is characterized by fibrosis and arteriostenosis, graft vasculopathy, which may be due to inflammatory reactions mediated by T cell cytokines. Graft rejection may be prevented by minimizing the immunogenicity of the graft by limiting the MHC allele difference and treated by immunosuppressions. Most immunosuppression is directed at T cell response and entail the uses of cytotoxic drugs, specifically immunosuppressive drug agent and anti-T cell antibodies. Widely used immunosuppressive antigen target calcineurins, mTOR, mechanistic target rapamycin, and lymphocyte DNA synthesis. Immunosuppression is often combined with anti-inflammatory drugs such as corticosteroids and inhibit cytokine synthesis by macrophage and other cells. Patients receiving solid organ transplant may become immunodeficient because of their therapy and susceptible to viral infection and malignant tumor. Xenogenic transplantation of solid organ from pig into human is limited by the presence of natural antibodies to carbohydrate antigens on the cells of discordant species that cause hyperacute rejection. Other mechanisms of xenographic failure include antibody-mediated acute vesicular rejection, T-cell mediated immune response to xenogenic MSC molecules, and prothrombotic effect of xenogenic endothelial on human platelets and the coagulation proteins. The ABO blood groups antigen are polymorphic carbohydrate structures present on the blood cells and endothelials that limit the transfusion and some solid organ transplantation between individuals. Pre-existing natural anti-A or anti-B IgM antibodies are present in individuals who do not express A or B antibodies on their cells, respectively, and these antibodies can cause transfusion reaction and hyperacute allograft rejection. Rhesus antigens are proteins on red blood cells that can stimulate IgG antibody response in Rh negative women carrying Rh positive fetus, and these anti-Rh antibodies can cause hemolytic disease in Rh positive fetus during subsequent pregnancies. Hematopoietic stem cells transplants are performed to treat leukemias and genetic defects restricted to hematopoietic cell. Hematopoietic stem cell transplant are susceptible to rejection, and recipients require intensive preparatory immunosuppressions. In addition, T lymphocyte and natural killer cells in the hematopoietic stem cell graft may respond to alloantigen of host and cause graft versus host disease. Acute graft versus host disease is characterized by epithelial cell death in skin, intestinal tract, and liver. It may be fatal. Chronic graft versus host disease is characterized by fibrosis and atrophy of one or more of these same target organs, and the lungs, and also may be fatal. Hematopoietic stem cell transplant recipients also often develop severe immunodeficiency, rendering them too susceptible to the infection. That marks the end of this chapter of transplantation immunology. I hope it was an interesting chapter and you have enjoyed it. And see most of selected things here for reading for you to enjoy more as we are preparing ourselves for tumor immunology as our next talk. All the best, Jeremiah Pusa.